Hello, my name is Carol Leibel, and we're going to examine the process of protein synthesis. Proteins are the workhorse molecule found in cells. Cells have many different kinds of proteins with many different functions. That being the case, you might ask, how do cells know how to make a particular protein? The blueprint or the instructions for the construction of different kinds of proteins is coded in the DNA of an organism. Humans have over 200,000 different kinds of proteins, and when you take into consideration all the different kinds of antibodies produced by the immune system, it's actually much higher. Proteins are made out of tiny building blocks called amino acids. There are 20 different kinds of amino acids, and when you string different amino acids together, you will make a polypeptide chain. The order that the amino acids come in a polypeptide chain is called its primary structure. The average polypeptide chain is over 400 amino acids long. Now you will notice in this slide that proteins are folded. Okay, these are two different kinds of models here. One is called a space filling model and the other one is a ribbon model of proteins, and that it's folded. In examining how a polypeptide chain is folded, you are looking or examining the secondary and tertiary structure of a protein. Now, some proteins actually have more than one polypeptide chain. In that case, you are examining the quaternary structure. Remember, genes produce proteins. This slide shows you some uses of different kinds of proteins. Okay, you are looking at red blood cells, and inside those red blood cells is the protein hemoglobin, which has attached to it iron, which causes it to be red. Iron has the ability to combine with oxygen, and so hemoglobin will carry oxygen from point A to point B in an organism. Those are enzymes. Remember, enzymes are biological catalysts that speed up a biochemical reaction without being used in the biochemical reaction. Enzymes control many biochemical pathways. In this egg, we have the protein albumin. Albumin is a storage protein that provides all the amino acids necessary for an embryo to go from to grow into a full-formed chick. That includes amino acids for the feathers and the beak and so forth. These are the proteins actin and myosin, which are interacting with one another to contract, and this happens inside your muscles. Muscles are used for movement. And if you look at the flagella of a sperm cell, inside that flagella are proteins. This right here are antibodies. Antibodies are produced by B cells in the immune system. Antibodies are used to fight off invading diseases. And what you're looking at right there is the silk produced by a spider. That's a structural protein. You also have structural proteins in your hair and nails, and there are structural proteins in the feathers of birds and the scales of fish and reptiles. And so let's look at the history of protein synthesis. In 1909, a British physician by the name of Archibald Gerard suggested that genes determine the phenotype through defective enzymes controlling biochemical pathways. He was investigating a human disorder that causes urine to turn black. He suggested that there was a gene that was not producing a particular enzyme that caused this urine to turn black. He collected family history and as well, of course, urine from his patients, and he was also familiar with the work of Gregor Mendel. Gerard deduced that this condition was a recessive disorder. In 1902, Gerard published a book called The Incidence of Alcapintura, a study of a chemical individuality. This was the first published account of a case of recessive inheritance in humans. And then moving on, there's the work of George Beadle and Edward Tatum, and they established a link between genes and enzymes, and remember enzymes are proteins, and studying bread mold. The scientific name for bread mold is Neurospora crassa. And you and I both know that bread mold will grow on just about anything. I have found bread mold growing on a cup of tea without any sugar on it. Don't ask me how that happened, but it did. So, at any rate, we have found that bread mold will grow in what we call minimal media. And minimal media is just a place for it to grow, agar. And in that agar, the bread mold is provided with simple sugars like glucose and fructose, and inorganic salts like sodium chloride and calcium chloride. And what is most amazing is that this bread mold can take those simple substances and make amino acids. They can string those amino acids together and make all kinds of enzymes and have sophisticated biochemical pathways occur inside the organism. So, investigating this simple organism, these two scientists took the bread mold and they exposed them to X-rays. Now, X-rays have been known to cause mutations in the DNA. And what they did next is they took this mold and they grew it on what we call complete medium. Everything that a bread mold could need, sugar and salts and lipids and amino acids were provided, and vitamins were provided to the bread mold. And so of course it grew. There was no problem. Then they turned around and they took spores from individual bread mold and they put it on the minimal medium. Okay? Now, if there was no growth, that told them that that individual bread mold had a mutation in it. And so then they started to studying different kinds of mutations. And so they found a biochemical pathway based on these mutations. The biochemical pathway that they investigated produced the amino acid arginine. All right? So this is what they did. They found out that this particular biochemical pathway would start off with hardly anything at all, and that the things that were produced along the way was this substance orthonine and citrulline, and finally the end product was arginine. So if you had some bread mold and the bread mold had no mutations and you grew it on minimal media, it would grow. If you added the other three things, well, of course it would grow. It really didn't need those things to grow. All right, well, then they turned around and they took this a mutant and they grew it on minimal medium, and guess what? Nothing took place. Well, then they turned around and they took this same bread mold, some spores from the same bread mold, and they had minimal media and they gave it orthonine. And it grew. Well, then they gave it citrulline, and it grew. And they gave it arginine, and it grew. So something was happening in an enzyme to go from here to here that it would not produce orthonine. Well, then they turned around and they found another mutant. And they tried the minimal media. It did not grow. Well, they tried it with, they added orthonine, and it did not grow. Well, then they added citrulline, and it turned around, and it grew. And then they added arginine, and it grew. So that told them that, hey, there was another enzyme involved, and we'll call that was enzyme A and that was enzyme B, to go from that place. Well, then they tried one last one. And they turned around and they tried it on minimal medium. And remember, this is a different mutation altogether. Nothing grew. Orthonine, nothing grew. citrulline, nothing grew. So that means that right now this last enzyme is the one that's defunct. But if you gave it arginine, well, then it grew. Okay? So what that told them, that in this biochemical pathway, that there were indeed 3 enzymes. And these 3 enzymes, we'll call them enzyme A, and you need enzyme A to produce orthanine, and then the fungi would take orthanine using enzyme B and produce citrulline, and then it would take citrulline with enzyme C and produce arginine. And that if you knocked out any of these enzymes, depending upon which enzyme you knocked out that you had a mutant in, it would stop the biochemical pathway in certain places. So because these biochemical pathways were controlled by enzymes, Beadle and Tatum came up with the conclusion that one gene would produce one enzyme. Now, the majority of enzymes are all proteins, but not all proteins are enzymes. And so therefore, this had to be modified so that one gene produces one protein. But as I mentioned, some proteins are made out of more than one polypeptide chain. And so it was modified to say one gene produces one polypeptide chain. Now here recently, in the last 10 years, it's been modified even further. Today, the definition of a gene is that it's a sequence of DNA molecules that can direct the synthesis of some sort of molecule product, in particular RNA. Not all RNA molecules that are produced are used as a pattern to make a protein. Some RNA molecules are translated, serve other functions used in gene regulation. Alright, so let's have an overview of protein synthesis. Some of the things we know in a eukaryotic cell is that DNA is located in the nucleus, and remember that's the genes. However, the proteins are actually made out here in the cytoplasm. That being the case, there has to be an intermediate, and it turns out that intermediate that is used as a template to take the instructions from the DNA out to the cytoplasm in the production of the protein is RNA. So let's compare DNA and RNA, because we know that both of these molecules are nucleic acids. Well, first off, we know that in most cases DNA is double-stranded and RNA is single-stranded. However, recently we have found instances where there are double-stranded pieces of RNA. RNA uses the pentose sugar ribose, and if I say it's a pentose sugar, what is its molecular formula? Think for a second. C how many, H how many, O how many? Okay, you're probably saying C5, H10, O5, and you are absolutely correct. Now DNA uses the pentose sugar deoxyribose, and think about the word decapitate. It means to take off the head. Well, deoxy means to take off an oxygen, and the oxygen that is missing is on carbon number 2. And so deoxyribose is C5H10O4. Okay? Now, it turns out that DNA and RNA uses adenine. They use guanine, which are both purines. They use cytosine. However, DNA uses thymine, but RNA uses uracil. And you can see the only difference is that uracil is missing a methyl group right there. but they're very similar. They're both pyrimidines. Okay. RNA can be found in the nucleus and the cytoplasm. In general, RNA is much, much smaller. DNA is restricted to the nucleus and organelles such as the mitochondrion and the chloroplast, and it is much, much longer. RNA has multiple uses, as we'll see. DNA is used as a template to make an RNA molecule, and that is used later on to make a protein. Alright, so there are three main types of RNA. However, as time goes on, we are finding that there are more and more uses for RNA. Okay, so first off, there is messenger RNA. Messenger RNA carries the information from the DNA gene out to the cytoplasm, and it's used as a template or a pattern to determine the sequence of the amino acids for a particular protein or polypeptide chain. Transfer RNA is responsible for bringing the correct amino acid to a ribosome and the messenger RNA in the process of translation. So looking at this, here's the transfer RNA. It kind of looks like a clover leaf, and then right up here is where we see an attached amino acid. All right. And then there's ribosomal RNA. Ribosomal RNA is found on the ribosomes, and it's used to loosely connect the transfer RNA to the messenger RNA in the process of making a protein. There's also something called small nuclear RNA, which is found on a structure called a spliceosome to remove unwanted pieces of RNA in a piece of messenger RNA. And then there's also small signal recognition particle RNA, which is used to help bring a ribosome to the endoplasmic reticulum in making proteins that are going to be exported out of the cell. Protein synthesis is a conserved biochemical process that is found in both prokaryotic and eukaryotic cells. All cells do protein synthesis. The thing about prokaryotic cells is that prokaryotic cells and prokaryotic cells do not have a nucleus. Okay? And so therefore, when the DNA is converted into, not converted, but when the DNA is used as a template to make messenger RNA, it occurs right into the cytoplasm. However, with a eukaryotic cell, it occurs in the nucleus. Once the messenger RNA is synthesized from the DNA template, then the messenger RNA in a eukaryotic cell is processed. You'll notice that this piece of messenger RNA is smaller and that there are these yellow parts, tails, and a cap that are added to the messenger RNA. Over here, no processing of the messenger RNA occurs. You'll notice that they are both the same size. Okay, and quite often, as this messenger RNA is being made, the polypeptide chain is being almost made at the same time. Okay, so transcription and translation can be simultaneous in a prokaryotic cell. That's not true of a eukaryotic cell. One of the things that they do have in common is that they use the same genetic code. Well, the question is, what do you mean by a genetic code? Looking at this diagram, one of the things I want you to notice is that here is a piece of DNA, and I want you to be sure you're aware that on a piece of DNA there are thousands of genes, and so this piece of DNA is continuing on. Okay, so let's look at this particular gene. Here is a sequence of DNA nucleotides, and that's the template, and it's trying to code for a particular polypeptide chain. And so this is going to be used in order to make a messenger RNA. Now you'll notice that over here, if the nucleotide is adenine, over here the complement of that is going to be uracil. If this nucleotide on the DNA is cytosine, over here on the RNA it's going to be guanine. So it's A for U and G for C. The other thing to notice is that 3 of these nucleotides form something called a codon. And it's going to give the code or the instructions for what amino acid is needed. So the DNA codon is ACC, the RNA codon is UGG. And what it's saying here is, I want you to make a polypeptide chain that has the amino acid tryptophan. Okay? And over here, here's the messenger RNA codon that says UUU, and that means I want that polypeptide to have phenylalanine, and then GGC, and I want the amino acid glycine, and then UCA, the amino acid serine. Alright, so now I want you also to understand why a codon is 3 nucleotides long. Whoops, let me go back. Sorry about that. I'm trying to erase this and I'm not doing a very good job. So, but let's just start then, okay? Let's suppose that if a codon was only 1 nucleotide long, okay? If a codon was only 1 nucleotide long, then you could only code for 4 amino acids. But let's suppose you use 2. If you use 2, you would have, here's your A, here's your G, here's your C, here's your U. This would be, you could have AAA, AG, AC, and AU. You could have GA, GC, GG, and GU. You could continue on, and I'm going to tell you right now, you could get 16 different combinations. Now, here's the problem. There are 20 amino acids. Well, 20 amino acids, that's not going to be enough. So it made sense that during the course of evolution, 3 codons would represent an amino acid. So if you had AA, now you could have AAA, AAG, AAU, and AAC. So for each one of these right here, you would have 4 more combinations. So 4 times 16, you end up with 64 different codons. So how did they ever crack the genetic code? So here's a little history on that. There were 2 gentlemen, Marshall Nirenberg and Heinrich Mattei, and their experiment was performed May 15th, 1961, and they cracked the genetic code because they took a test tube, and what they put in that test tube were amino acids, ribosomes, RNA polymerase, and other needed materials. And then what they added was a piece of messenger RNA that contained only uracil, UUU, UUU, UUU. Okay? And then they looked at what was made. Well, it turns out that the codon UUU codes for a particular amino acid, phenylalanine, and so all they got was phenylalanine, 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 and so forth. And so what that told them is that the codon UUU coded for the amino acid phenylalanine. Later, Nirenberg worked with a gentleman named Philip Later, and they determined the triplet nature of the rest of the amino acids. And so they made some discoveries about the genetic code. what they found out is that the code has redundancy. For example, look at this. CGU, CGC, CGA, and CGG all code for arginine. And in addition, AGA and AGG also codes for arginine. So there's a lot of redundancy in the genetic code. Remember, there are 64 codons, but there are only 20 amino acids. All right? Now, there are also 3 stop codons. So actually, 61 code for amino acids. And then down here, this is kind of interesting, AUG codes for the amino acid methionine, but it also codes for start. It turns out that each codon codes for only one amino acid, or start or stop. The code is universal, meaning that almost all cells use the same genetic code, prokaryotes and eukaryotes. There are some exceptions, and here are a few exceptions. In the animal mitochondria, uses AUA for methionine instead of isoleucine, and for vertebrate mitochondria, AGA and AGG are also used as stop codons. And in the paramecium, where UAA and UAG represents glutamine instead of stop in a species of paramecia. So there are a few exceptions, but not many. Alright, so here's an overview. Here's a piece of DNA, and this is going to be, whoops, let's go back, sorry about that. Alright, so this is going to be a DNA gene, alright, and these are the codons. And so RNA is always made 5' to 3'. And so this right here is going to represent the side of the DNA that's going to be transcribed, because that will be, of course, 3' to 5'. And so when we transcribe it from DNA into RNA, meaning the DNA is used as a pattern or a template to make this RNA molecule, then this RNA molecule is going to be used as a template or instructions to make this protein. And the protein, using these codons, AUG represents methionine, GAG represents glutamine, CAU represents histidine, and CCC represents proline. And then UAA means stop. That's where the protein will stop. So ultimately, this gene says I want a protein made, and I want the protein made so methionine comes first, glutamine, histidine, proline, and then stop. That's what the gene or the template is saying. I want this particular polypeptide made in this particular order. Alright, so this is an overview of transcription. We're going to get down to the nitty-gritty. Okay, remember, transcription means that we are taking and we're going to make RNA using a DNA template, and there's initiation, and that's where the RNA polymerase is going to attach. Then there's elongation, where now we're making the RNA molecule, and then there's termination, where the process stops. Once the RNA is synthesized, if you're talking about a eukaryotic cell, then the RNA has to be processed. Alright, so in initiation, if you're looking at the gene, there's a region prior to the beginning of the gene. Okay, there's a region prior to the beginning of the gene where the RNA polymerase attaches, called the promoter region. And the promoter region is going to determine which side of the gene is going to be transcribed. And it's always going to be the side that is 3' to 5'. In a prokaryotic cell, the RNA polymerase attaches directly to the region, but in eukaryotic cells, what has to happen is that there are going to be some transcription factors needed in order for the RNA polymerase to attach. Okay, within that promoter region, there's a sequence of thymine and adenine nucleotides right here. And it's called a TATA box, or I hate to say it, a TATA box. And that identifies where the RNA polymerase is going to bind. And once it attaches, like I said, even more transcription factors are going to attach. And now the RNA polymerase is going to unwind the DNA and start base pairing the DNA to the RNA gene. In prokaryotes, there's only one type of RNA polymerase, but in eukaryotes, there are 3 different types of RNA polymerase. Each one will produce a different type of RNA. So the next step is elongation, and during elongation, you can see that the RNA polymerase is starting to base pair, and it always goes from 5' and we're adding on to the 3' end. And you can see that the base pairing is, if there's a thymine on the DNA, you'll add adenine. If there's an adenine on the DNA gene, you'll add uracil. Okay, and the nucleotides are added to the 3' end. Okay, and as the polymerase then moves on, the RNA peels off and the DNA will reform its hydrogen bonds. Alright, so now termination. In termination, what's going to happen is the RNA synthesis proceeds until the RNA polymerase encounters a sequence that's going to trigger the RNA to, basically the RNA to fall off, the RNA polymerase and the RNA to dissociate. In eukaryotic cells, the RNA polymerase will actually pass this termination point before the RNA molecule is released. In prokaryotic cells, however, there are 2 things to take into consideration. There's what we call the intrinsic termination, and the RNA transcription stops when a newly synthesized RNA molecule forms a GC-rich hairpin loop. So what happens, the RNA is falling off, and then right in here we see this hairpin loop of guanine and cytosine, where it will actually they'll form these hydrogen bonds. And now what's going to happen is that mechanical stress will break the bonds, and it will pull off the RNA polymerase, and that will indeed terminate transcription. And then it turns out there's an extrinsic factor, and that means that what happens is that what attaches here is a protein called a Rho protein, and as it goes through, and once it hits this area right here, that will also cause the RNA polymerase to fall off. And that, my friends, causes the end or the termination of transcription. Alright, so here is a nice little video on transcription. It's coming from the internet.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
All right. And I'm sure it did a much better job than I did, but I hope that makes sense in how transcription is terminated in a prokaryotic cell. All right, so let's get back to eukaryotic cells, because one of the things we mentioned about a piece of RNA that is made from a DNA template in eukaryotic cells is that it's processed. One of the things that we know is that the average immature messenger RNA that is comes off a DNA template is 8,000 nucleotides long. However, when that messenger RNA goes out into the nucleus, it's only 1,200 nucleotides long. And, well, 1,200 makes sense because remember I said the average protein is 400 amino acids long. Each one has 3 codons, so 3 times 1,200 nucleotides long. But originally it was 8,000. So the question is, what in the world happens? How did it get so small? Well, it turns out in eukaryotic messenger RNA, quite often there are areas that are not going to be expressed or used. Those are called introns, and those are going to have to be removed. In addition to that, at the 5' end, we're going to add what we call a 5' cap. And in addition, on the 3' end, we add a series of adenine, 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 and that's called the poly A tail. And so once you remove those introns, then what's going to happen is what's remaining are the exons. And now you have a mature messenger RNA. The poly A tail can be 300 to 200 adenines long, and the 5' cap varies from messenger RNA to messenger RNA. And it is thought that the messenger RNA needs that cap in order to be able to move through the nuclear pore of the nucleus. Alright, so now the question is, how do you get rid of these introns? Well, that, my friends, involves using something called a spliceosome. So let's watch this nice little animation about using spliceosomes.
Most eukaryotic genes are composed composed of numerous short coding sequences called exons, embedded within stretches of non-coding sequences called introns. The initial messenger RNA molecule, or primary transcript copied from a gene by RNA polymerase, is a faithful copy of the entire gene, including introns as well as exons. Before the primary transcript is translated, the introns are removed by a process called RNA processing, or RNA splicing. Particles composed of proteins and a special type of RNA called small nuclear RNA, or snRNA, play a role in RNA splicing. One kind of small nuclear ribonucleoprotein, snRNP, contains snRNA that can bind to the 5' end of an intron by forming base pairs with complementary sequences on the intron. A different snRNP binds to the 3' end of the intron. Additional introns interact, causing the intron to form a loop, thereby bringing the two ends of the intron together. The large complex of snRNPs, called a spliceosome, then excises the intron and the exons are joined together. The snRNPs are then released.
So let's look at this. Okay, again, here we have an intron, and here we have some small nuclear RNA and some other proteins. What's going to happen is they're going to come together and form something called a spliceosome. The spliceosome is going to bring the ends of the exons together and join them and basically cut out this, they call it like a lariat of RNA. Okay? Alright, so an important thing that has to be done in processing eukaryotic messenger RNA. Now, here's a very interesting thing. They have found that humans have between 23 and 25,000 genes. Yet I mentioned we can make over 200,000 different kinds of proteins. How in the world does that happen? One of the things that they are now aware of is called alternative splicing of the messenger RNA. So here we have some messenger RNA, and in one case this intron is removed and that intron remains. And so translation takes place and you have that sort of a protein. Over here, in this case, this intron is removed, but that one remains, and so now we have a different protein. Alright? And so by alternative splicing of the RNA by the spliceosomes, you will be able to make different kinds of proteins. Extremely important in producing different kinds of proteins with a smaller number of DNA genes. Now, here is a gentleman that's going to tell us about alternative splicing.
In our body are expressed as RNA in different cell types. But in one cell type, the pattern of splicing of the synth pieces together is different than the pattern in another cell type, and that's called alternative splicing. So it's alternative splicing because the splicing pattern in one cell is different than the splicing pattern in another cell, and therefore the proteins in those two cells are different. And that can make a great deal of difference because one protein can function spliced in one pattern, in one cell one way, and then another spliced differently in another way. So if you're designing drugs or you're looking at functionality and mutations, they can differ due to this alternative splicing.
Alternative splicing means that the splicing machinery selects certain exons or parts of certain exons differently in proportion to specific. So it's a way to take a gene and from its primary transcript to generate two
Well, I hope you were able to hear that. I'm sorry it was a little bit, the volume was down a little bit on my side too, but at any rate. So one other thing I want you to look at that will produce different kinds of proteins, and this has to do with the DNA gene itself as opposed to alternative splicing of the messenger RNA. Proteins often have what we call like a modular architecture consisting of discrete regions called domains. And in many cases, different exons will code for different domains in a region. And there's a domain shuffling, if you will, to produce different kinds of messenger RNA, which ultimately results in different kinds of proteins. This exon shuffling and rearrangement of these domains happens in the productions of different kinds of antibodies in the immune system. And this will also allow you to have fewer genes but yet have a greater number of proteins. Alright, so the last bit. Now we are finally ready for translation. The messenger RNA has been processed. Introns have been removed. We've added a 5' cap and a poly A tail. In order for this piece of messenger RNA to go out to the cytoplasm, it usually has to attach to some sort of protein signal that will allow it to move through the nuclear pore. It is known that not every piece of mature messenger RNA that is made in the nucleus ever reaches the cytoplasm. Some of them are unable to cross the membrane of the nucleus. Alright, but once it reaches the cytoplasm, folks, translation is about to occur, and that's the next PowerPoint. So get ready to make that protein and translation. Let's go. Take care. Bye.